Hi guys, today we'll be looking at the differences between mitosis and meiosis. To do this though, we must look at what both of them are and how they work. Mitosis is a process which happens in somatic cells and is a process which makes two daughter cells that are exactly identical to the parent cell. This doesn't just happen all at once though, and there are steps that are part of mitosis. The first stage of mitosis is interphase. You can see in the top left rectangle of the picture. To sum up what interphase is, it's basically the cell preparing to divide. So it grows in shape, replicates its organelles, and replicates its DNA. From there, it is ready to go into mitosis stage. In mitosis, seen on the right-hand side of the picture, it goes through 4 stages of mitosis. First being prophase. In prophase, the pair of homologous chromosomes line up in the middle of the nucleus. Next, in metaphase, the spindle fibers attach to the chromosome centromeres and get ready to pull them apart. From there, it goes into anaphase. In anaphase, the spindle fibers pull the two sister chromatid apart and start to pull them towards the two opposite ends of the cell. The final stage of mitosis is telophase. In telophase, two nucleuses form, but the nuclear membrane is still intact. The last step of all of this is cytokinesis. In cytokinesis, shown in the bottom left-hand corner, the nuclear membrane is breached and the cell splits into two identical copies of the original cell. The other type of cell division is meiosis, and this happens exclusively in gamete cells, sperm and eggs for humans. As with mitosis, there are multiple stages to meiosis. And in this cell division, it will take a diploid cell, or a cell that has two copies of each of our 23 chromosomes, and make 4 unidentical cells that each have one side of their chromosome, otherwise known as a haploid cell. This and a couple other steps in meiosis is why we see genetic diversity in humans. First off, in meiosis, it is also started with interphase as with mitosis. The cells get ready for the rest of the cell cycle. It does this by replicating its organelles, growing in size, and replicating its DNA. When all of this is done, we move into meiosis 1 or M1. The first part of M1 is prophase 1. In prophase 1, the homologous pair of chromosomes line up in the middle of the cell. When the two chromosomes pair up in the middle of the cell, it is called a tetrad. Something interesting that can happen in prophase 1 is independent assortment. So basically what this is when the chromosomes are lined up in the middle of the cell, their homologous paired chromosome is the same number chromosome in the karyotype. One of the chromosomes from that number is from mom, and the other one is from dad. When they line up in the middle of the cell, though, the chromosome from your mom and your dad have an equal chance of being on either side of the cell. So in this case, with 4 chromosomes, there are 2 different options to what the chromosomes can line up due to independent assortment. This is one of the ways meiosis leads to genetic diversity. Another phenomenon that creates genetic diversity in prophase 1 is crossing over. Crossing over happens when homologous chromosomes come to the middle of the cell and actually exchange some of their genes with each other. There are a ton of ways that the homologous pairs of chromosomes could exchange genes in this crossing over stage. This is just another way that genetic diversity is created through meiosis. The next stage of M1 is metaphase 1. Not much special to this stage, and it is essentially the same as metaphase in mitosis, where the spindle fiber attaches to the centromeres of the chromosomes. Next, there's anaphase 1, where the homologous chromosome pairs are split apart to opposite sides of the cell. Then telophase 1 happens, and 2 nuclei are formed, but the cell's outer membrane is still intact. Finally, the cell goes through cytokinesis and splits into 2 cells. The most notable thing about M1 is the cell goes from 1 diploid cell to 2 haploid cells with only 1 copy of each chromosome. And this is very important because if this didn't happen, every generation in a population would have double the number of chromosomes that their parents had. So the next step of meiosis is meiosis 2 or M2. And M2 is exactly the same as mitosis, as you can see in my diagram. So I'm not going to be saying the same thing over again, but something to note is that with the whole process of meiosis, the goal is to make gamete cells genetically diverse and give each cell 1 copy of a chromosome. In this, we go from 1 diploid cell to 4 haploid cells. So now that we have looked into what each of the 2 cell divisions are, we can get back to the original question of what are the key differences between mitosis and meiosis. The first key difference is the ploidy of each of the parent and daughter cells. In mitosis, you're going from a diploid cell to make two diploid cells. However, in meiosis, you go from one diploid cell to four haploid cells. Another difference is the number of divisions that happen in each of the cell processes. In meiosis, there are two cell divisions of the cell, but in mitosis, there is only one. Probably the most important difference is in mitosis, you are making cells that are genetically identical to the parent cell. But in meiosis, because you're making sex cells, you make genetically unique cells. This is very important because it wouldn't be evolutionarily optimal for the offspring of organisms to be genetically identical to their parents. The type of cells that these types of cell processes happen in is also another difference. In mitosis, it happens in somatic cells, and in meiosis, it happens in gametes. Finally, mitosis is for growth and repair, whereas meiosis is for reproduction. So thank you for watching this video. It was a super fun video to make. Kind of going over the differences between mitosis and meiosis, and hopefully all of you guys learned something.